this is just so that you can get to know like us a bit better. So you're definitely going to know the lecturers when you start. Well done. And it's just to remind you of some of the things that people would have covered before starting. I guess the curriculum might have been different for you as well. So it might be useful anyway in that sense for you to see. So let me share my screen and then we'll see if you can use the stampers. Here we go. I need to get that one and I need to go back to here. Oh, okay. There we go. And it's nice because I can still see you as well. If you have any questions, Nivedita, just please either type it on the chat or stop me because it's just us. So this is a great opportunity for you. Take advantage of it, please do. I'd love to be able to just chat with you about it. That's much more exciting than me just talking at you anyway. So you might have seen, hopefully Alex told you last week about our Thinkific site, and you probably had to go to that to find this link anyway. So there is the catch-up compendium as well if you want to have a look at any of this. So you would just be able to have a look there as well. But this isn't meant to stress you or for you to revise or learn. This is just a get to know us. So don't feel you have to be stressed by that. Okay, so stampers, I'm hoping it's going to let me use. So you should be able to go to the top of the screen and there should be a pen that says annotate. Can you see that?
Yes.
And if you click on that, then you will find a tick that will say stamp.
No, I'm only getting something that says pen or highlighter.
You're only getting, so perhaps you can do a tick with the pen if you don't get the stamps. I don't know if Peter gets the stamps, but I get some stamps that look like this, so I can tell you that I studied biochemistry. But perhaps you can do a tick as to which one you're registered for. Excellent. Peter, have you got some stamps? I don't know if Peter's...
I am, unfortunately, I'm on an iPad and it doesn't look like I have this option.
Oh, well, there's some really fun stamps. I can demo them for you. We've got some stars, arrows, whatever you like. So, yeah. So cool. How about the nervous system? Did you cover that, Nivedita? Did you cover it? Do you feel like you've covered it a lot already? Or I've had to do loads because I've been teaching it.
The thing is, I did biology for my O-levels, but then for my A-levels, I did physics, chemistry, maths. So I had a long break from biology and all of this. But we covered the nervous system in the O-levels, yeah.
Yeah, okay. Oh, that's really good then. So today might actually be quite useful for you, hopefully. That's great. Okay. So yeah, it's a shame that this doesn't work on, I thought this stampers worked on iPads as well, but obviously not. So now I have to make it clear the drawings away, and then I should be able to close that and move on. Oh, it's not going to let me move on. This is the problem when you do these fancy things. There we go. All right, so there would probably, this is my favorite poster of myself. We all have to have a favorite poster of ourselves. So I just thought I'd at least tell you about me. If I'm going to be telling you things, you want to know that I'm qualified to do so. So I actually did do my undergraduate at Kent too. I did it in biochemistry. Peter taught me as an undergraduate. So I remember his lectures very well. I can tell you they're exciting and fun. And then I went away to Cambridge and I did a PhD in clinical pharmacology. And there I was learning quite a lot about the heart, and I was working particularly on druggable targets in the heart. So I was working a lot on that. And then I came back to Kent. By some luck, there was a job advertised. I thought I was going into industry actually, and I was mainly looking at industry jobs, but I'd had a really happy time at Kent and there was a job advertised. So I just thought, okay, I'll try that. And since then I've worked here in the biotechnology field. And within the biotechnology field, I'm really interested in protein translation, so how we make proteins. And that means I've become a bit interested in cold shock, and that means I've become interested in the nervous system. So it was a bit convoluted how I got interested in the nervous system. But that's how it happened. And And I'm a lecturer now in microbiology, and I'll teach you quite a lot at stage 1. I'll teach you quite a lot of physiology. I'll teach you quite a lot of molecular and cellular biology. So you'll see me a fair bit in your first year. So hopefully that's not a terrible thing. I should have also said, Navdeej, that this is being recorded. Are you okay with this being recorded?
Yes, I'm fine with that.
Okay, sorry, I should have said that at the beginning. Okay, so I will talk more about this at the end, because I'd like to just tell you a bit about our research as well, because it's kind of boring for you if you don't get to hear what the fantastic, amazing things that we're doing are. But as I said, the reason I got interested in the nervous system is because I'm interested in medical applications of the cold response. So obviously, if you go and get outside and get hypothermia and get very cold, there are responses that happen within your body. And this over here, this wonderful machine, let me get my laser pointer here, is my favorite machine in the Science Museum. And it's actually called the profound hypothermia apparatus. And it was actually one of the machines used for the first heart surgeries. And what they used to want to do is try to perform a heart surgery, but they didn't have bypass machines. So to stop people getting damage to the tissues, they used to try to cool them. And they realized that if they cooled them and tried to bring the blood out that way, it kind of went thick and gooey, so they couldn't work with it very well. So they actually contacted some manufacturers of ice cream. And the ice cream manufacturers came up with this awesome machine which would allow them to cool the blood and pump it through, because they were used to dealing with something of that consistency. So if when you're in London, you get to go to the Science Museum, go and have a look for this machine, because I think it's pretty awesome. But part of why they were doing that was to protect people from brain damage as well. So we know that if you cool premature babies when they're born, if they've been subjected to hypoxia, then it can protect them from brain damage. And we know if you cool organs for transplant, it protects them from damage. But something else that we know is that when animals hibernate, it protects their neurons to have this cooling effect. And we've started to understand a little bit about how this might relate to dementia and how this might relate to people who do cold water swimming. And I'll talk to you a little bit more about that later when I talk a bit more about my research. But basically, I got really interested in how cold therapy might be used to treat dementia. And one of the things would be, you don't really want to make elderly people be subjected to hypothermia, because that would probably kill them. So instead, what we want to do is find a pharmacological way of doing that. Okay, so that's why I teach the nervous system, because I've got some experience in working in that. So that's where my interest comes from. So let's give you a quick overview and a quick reminder of some of the things in the nervous system. So nervous system is organized into 2 parts. We've got the CNS over here and the PNS. So CNS is central nervous system, and central nervous system will be your brain and your spinal cord. So it's those core parts. And then we have the peripheral nervous system, which is pretty much just everything else that you've got there. And so you just basically need to know that there are those 2 subdivisions that are quite important in your nervous system, and to be able to understand that the central nervous system and the brain is the brain and the spinal cord, and the peripheral nervous system is everything else. And that's hopefully not too tricky, that part of it. The scientists don't really like to keep it that simple. We don't like to keep anything that simple. So of course there's loads more divisions than that. And these are all things that would have been covered sort of at A level. So the central nervous system is the brain and the spinal cord, like I said. We then have the peripheral nervous system. I've written CNS here. That should say PNS. Apologies for the typo on my slide. So that's nerves that connect all parts of your body to the brain. So first of all, here we've got your sensory division. So this is what's feeding in. This is what's feeding in. So basically, we have to be able to respond. Your nervous system is pretty awesome. It's going to help you to respond to anything that's coming in. So I don't know, tiger jumps in the room now, I'm going to be able to respond. appropriately to, if somebody walks in the room and taps me on the shoulder, I don't want to respond in the same way. But we have our sensory division to be able to respond to external stimuli, but equally we have to be able to respond to things internally. And so we also have input from our internal organs into our PNS as well. We also have something that's called our motor division. And so that's going to be carrying those signals back out. So the sense ones have put that in, carried signals in, and then the motor division is going to be carrying that back out. So it's going to be carrying the signal out to the CNS. Okay. If anything doesn't make any sense at any point, Nivedita, please stop me and just say.
Sure.
Okay, brilliant. So the somatic nervous system, that's the bit that's all our conscious activity. So if you do go and play a video game, I don't know, I don't actually play many video games, I kind of don't get time. But if you do play a video game, or me controlling Zoom now, then that's our conscious nervous system. So that's our somatic nervous system. And then equally, we have our autonomic nervous system. So that's all the things that you don't have to think about. Thank goodness we don't have to think about digestion. We don't have to think about controlling our heart rate. We don't have to think, normally don't have to think about when we're breathing. Sometimes we have to think about when we're breathing, but normally not. So have you heard of fight or flight and rest and digest before?
Yes.
Yeah, you have. Excellent. So there are sympathetic and parasympathetic nervous system are the other bits that you'll need to know. So our sympathetic nervous system is that fight or flight. So if the tiger jumps in the room and I need to do something, then that's my sympathetic nervous system that's going to kick in. My parasympathetic nervous system, on the other hand, is my rest and digest. So that's what's going to be kicking in when I can just kick back, absorb my food, relax and enjoy life a bit. Hopefully no tigers jumping at me at that point. So that's all the divisions of the nervous system. And when you get into stage 1, we'll look at those in a bit more depth, at what they mean and how they work and what the nervous signaling is within those. But this is just a reminder of those now. So all of those things, all of those divisions of the nervous system, they're all being carried out by, the signals are all being carried by neurons. And so another quite basic level thing we have to know is the structure of a neuron, and that's really important for lots of things to be able to understand the structure of a neuron. And again, actually we cover this a bit at stage 1, but it is covered at A level 2, and we just try and go a bit further and a bit more in depth once we get into stage 1. But for now, when we're thinking about just refreshing this, so we would have a nerve cell, and it has a nucleus like any other cell. But it also has these dendrites here. So these will be taking information in, and it will have this quite big cell body here. And on it, it has something we call an axon hillock. You don't need to worry so much about this, except to say it's where signals essentially start. And they'll be passed down this axon, and that will then go to the end to these synapses where it will communicate with other cells. Now, the problem is that if you just send electrical signals, it would be like if you just sent it down an electrical wire that wasn't insulated, and you've done physics and chemistry, so you'll know it will just dissipate, and it's not going to be a very effective mechanism of trying to transfer a signal. So neurons are myelinated, and this is a ring of a myelin sheath that goes around, and it essentially insulates the axon. And basically what it means is that the conduction happens much faster, and the conduction jumps from one gap to another gap. And these are called the nodes of Ranvier, these gaps. And so that makes it much quicker. If it can jump, the signal can jump from here to here to here, it means it's way, way faster than it would be if we just had this as an unmyelinated axon. So the myelin sheath is really, really important. The cell body is really important because all the things that the cell needs have to be made here still. We still have to have transcription and translation and making proteins and all those things that, again, we'll cover in much more depth at stage 1 of your degree. And I'll be teaching you transcription and translation as well, actually. Okay, so this is where I will ask you to do a tick again if you can, or you could use a stamper if you were able to access it, but such a shame you can't. So what do you think? What do you think might affect the speed of conduction? Myelination, the diameter, the temperature, none of these or all of these? Temperature, yeah, okay. So the actual answer is all of them. So yeah, I think that you're I can see the physics here coming out. I can see you definitely studied some physics, so that's great. So I would yeah, be putting my stamper down here. But yeah, all of these things can actually have an impact on the speed of conduction. And it's great. So there's some giant squid actually that have these massive giant axons, and they aren't myelinated, but because they're so wide, the conduction still can occur quite well. So all of those things actually can affect conduction, but myelination is one that we think of really importantly for a particular disease. So do you know which disease might be affected by myelination?
No.
No, okay, that's fine. Lots of people haven't heard of it and some have. So I just thought I'd check before. Oh, I'm just carrying on doing stampers there. Look, let's get rid of those so that we don't sit there with them on there. Okay, good. And let it come on then. So actually, it's the answer is multiple sclerosis. So again, what we do at stage 1 is we have quite a big focus on different diseases within your nervous system. I'm not going to cover all of those today because that's what we're going to cover at stage 1, and then you'll be really bored when you do your degree if I try to cover it all today. But we'll cover things like dementia, Alzheimer's disease, Parkinson's disease. We'll look at different types of myelination diseases. So there's all sorts of different types of those. And one of those that we'll be looking at will be multiple sclerosis. And so multiple sclerosis is exactly what you can see in this picture here. Here's a normal nerve fiber with its lovely myelin sheath on it. But in multiple sclerosis, which is actually an autoimmune disease, you start to see a degradation of this myelin breaking down. And because that's happening, basically it's going to affect that speed of conduction. So if it affects the speed of conduction, it's going to cause us all of these different problems here. Oh, this box is completely blocking this for me. Let's move that. It's going to cause all of these problems here that we'll have typically in MS. So things like blurred vision, it causes CNS problems. So it can be like dizziness, hearing loss, fatigue. You can have difficulty swallowing and difficulty with speech as well. It causes changes in your digestive system, so it will cause constipation, diarrhea, senses. So typically people think of MS, they think about pins and needles. That's one of the signs that people might look for, is pins and needles. And you can have cramping too. And it's really an interesting condition actually, because your myelin sheaths can actually regenerate a bit. So kind of depending on what stage you're at within the autoimmune part of this, you can have different types of MS. So we can have a relapsing and remitting MS. So one that comes and goes, and people can be quite well for many years like that. You can then have one that then, once it's been like this for a few years, starts to just continuously progress, so secondary progressive. Or you can have primary progressive MS. So that's unfortunately a type that's just aggressive from the beginning and continues to be that way. So it's why you might hear of people with MS who have very different condition, are in very different physical condition, and actually it can really impact how life expectancy and those kind of things. And of course we do have lots of treatments for those now. So something I'm really interested in is making antibodies in cells. So we have big research groups here, led in particular by Professor Mark Smales, and his research group are really interested in making monoclonal antibodies in cells, and you can make antibodies which are used to treat conditions like MS actually. So that's really exciting research that's happening here, and hopefully you'll get a bit of a chance to see that when you come, get to see the research labs a bit more as well. Okay, So being knowing a bit more about physics, do you know much about membrane potentials or not really? Shall I go from the beginning?
I know a bit about it.
A little bit. Okay, good. So I won't go too far from the beginning then, but basically the membrane potential is something that every cell will have. It's a way that we depict the charge, the voltage across the cell membrane. And all cells in your body will have this, and that's because they all have ions within them, and we have channels and pumps within the cell membranes that are able to affect this. So what we've got here is just an example. This is depicting a cell membrane. We've got ICF stands for intracellular fluid and ECF stands for extracellular fluid. And then we've got the different ions here that we might typically find in and around a cell. So sodium ions, chloride ions, we have potassium and anions. And so basically within a cell, generally, if we didn't have any pumps or any movement, then as long as everything was balanced and equal, this could be at 0. But that's not what happens because we do have pumps and we do have movement of ions, so everything's not at 0. And the main reason for this, the main sort of contributors to membrane potential, I would say, are the sodium potassium pump. So that's this one here. And the sodium potassium pump is basically pumping out 3 sodium ions for every 2 potassium ions it pumps back in. And it uses energy to do it. So it uses a molecule called ATP and it uses energy to do this. And so this you can instantly see is creating a charge balance difference because we have 3 positives coming out and only 2 going back in. So already that's creating a negative balance difference there. We also have something we call potassium ion channels. So these aren't working in the same way. They're kind of, they're not requiring energy in the same way. So this movement can be happening all the time. But what it means is that in the end, in cells, everything balances out to have a negative resting potential within the nervous system in particular. This can vary in different cells, what this number is. So in the nervous system, it's -70. If you're looking in the cardiovascular system, this could be -55. So this can vary depending on what channels and receptors you have in the membrane. But in the nervous system, the typical value we would give would be around -70. But I won't go much more into ions, seeing as you already know that and you've done lots of physics. It would be a bit boring for you if I start trying to just lecture you on ions. So I think we'll just, as long as that's okay with you, I won't keep boring you with the detail of that. Okay, so here's a challenge. I'm terrible at drawing. I'm just going to confess now, my hand is not neat at this. So this is a kind of a challenge for me. Would you know what an action potential looks like at all? Can you remember it at all?
No.
No, it's fine.
I don't think I've heard that term before.
Okay, excellent. Oh, good. So we can go through what an action potential is as well. So it's something that you haven't covered perhaps so much. Brilliant. So an action potential is the way that a nerve cell basically sends its signals, and it's a way of conducting a signal all the way along the nerve cell using that difference that we were talking about here, using the potentials. So basically, I'm going to turn on my pen. I'm going to try and draw this. I might make a rubbish job of it because, as I say, I've got a really unsteady hand. But if not, on the next slide, I have a lovely, neat drawing of it that will look much better for you. But I always think it's better if I'm at least trying to do this as we go. Okay, so we have that potential here, -70. And this is basically, oopsie, I'm really wobbly. This is basically our resting potential, and it'll be sitting somewhere here. Oh, is it going to keep disappearing? I don't want a vanishing pen. Give me a normal pen, draw. There we go. Let's do one that doesn't vanish. Okay, so it's basically sitting along here. Okay, so let's imagine it's me being tapped on the shoulder. So basically someone comes along and taps me on a shoulder, and that's going to activate receptors in my sensory system to say, oh, there's a signal here. We need to pass this signal on, something's happening. So at this stage, we're going to have an event which is called depolarization. And this depolarization occurs like whoop, up here, like this. So basically a depolarization means that we're getting into being more positive. So there's a more positive ion charge going across the membrane. And how is that happening? We'll talk about in a minute. So, but first of all, I just want you to get the depolarization, repolarization parts in, but it's to do with those sodium channels and those ion channels that I talked about before. So basically we've got movement of ions across, and that's causing us to go more positive on the membrane potential. So that's the depolarization. And then what happens is we have some more channels that open, and on the next slide where I'm a bit neater, I'll show you those. And we get this repolarization. So it is just like you might see in physics, like a spike, basically, of electrical charge that's then going to need to be carried along an axon. And the way we make that spike of electrical charge is by all the ions. And we then get something that happens that's really important. It overshoots before it gets back to resting potential here. So this is essentially an action potential. And if you just sort of done biology more recently, then hopefully you might have been able to draw that, but that's fine that you can't, because that's great that you're at this catch-up session. So that will help you next term, at the first term, hopefully. So if I show you it more neatly, because this is not neat the way that I have done this, turn annotate off, and I can show you it with the ions on there as well then. Oh, there you go. So you can see how awful mine was. So let's just clear mine. I was interested to see. So let's do this. That's much neater now. So here you go. This is what you can see. So we've got a nerve cell at rest. A stimulus came along. It caused depolarization. How it did that was sodium channels opened. So the sodium channels opened, and it meant that more positive sodium was able to move into the cell. So that happens because if we go back here, as well as needing to balance out charge, we also balance out essentially across concentrations. We have two things that will contribute, and you can see there's many more sodium ions outside of the cell, and also the inside of the cell is negative. So if you open sodium ion channels, they're all going to try and flood into the cell just to try and balance out the charge and balance out the ion concentrations. So that's how that's happening. Once we get to here, they open, and that makes this positive spike of voltage. You can see it's measured in millivolts here. And then at some point, those channels close. So enough has come in and they close. And meanwhile, now some potassium channels open. These potassium channels have a really neat mechanism that we'll go into at stage 1 about why they open more slowly than the sodium channels. But for now, that's all you need to know, is they open more slowly. And that allows the potassium channels now to be moving. And that allows a repolarization, because the potassium can move back out and balance all of that charge that's just come in. And that allows repolarization, so the cell has this spike of activity, and then it goes back into something that we call hyperpolarization. And finally, it'll go back to its resting, ready to get a new signal. Now, this hyperpolarization is really, really important, because what it does is it stops your nerve cells getting loads of signals all at once. It would be no good if your nerve cells just had all those signals happening all at once. So while it's in this phase, you can't get another signal coming in and activating just in this brief point. And that stops this summation. So basically, you don't want your nervous system just turned on constantly. That wouldn't be good for you or me. I mean, everything we've done this morning has been controlled by our nervous system. And if it was constantly on and firing, that would be pretty uncomfortable for us. So we also have to have this little period here that it overshoots before it gets ready to go and take another signal. So that's an action potential. So that's essentially what's going to be leaping along that myelin sheath and the axon inside the myelin sheath that I showed you earlier. And it's happening like a domino effect. So basically, I like to think of it as a whole wave of dominoes, and they will literally move along the action potentials all the way along the axon. And that's how an electrical signal is carried along. Does that make some sense? Do you have any questions about that?
No questions.
No, it's quite a lot to take in the first time, I know. So hopefully you'd be able to go and watch this video back. But I think I would say you can probably find this in the Thinkific guide, but it is just like literally knocking over a whole heap of dominoes, one after another. They fire along that axon. But I don't want to, again, we go into that a lot more at stage 1, and I don't want to overcomplicate it now. But at A level, this is what would be learned, that this happens in order to trigger that. Okay. So that happens. So we had that cell. Let's go back and just show you that. So that happens. Basically, that action potential happens, and it will go all the way along, firing the next one, the next one, the next one. It triggers all the way along. It happens in milliseconds to get to the end of here. And once it gets here, that's great. We've fired a signal along a nervous system. Okay, so the cells I've touched here have fired a signal along the first part, say, to my spine. But now things need to communicate. It's no good just firing it along one nerve cell. They need to be able to communicate. So the way that nerve cells communicate is here at the synapse. They talk to each other using a synapse. Now, synapses can be electrical, but most of them are chemical. So we're just going to talk a little bit more about those now. Okay, so this is a synapse. This is the bit where they talk. So this is that bit that I just showed you, and this is where they're going to talk to one another. So here we've got one of those synaptic terminals on the end of that nerve cell that I just showed you. And it has chemicals in it. And those chemicals are called neurotransmitters. So hopefully you've heard of neurotransmitters. They're pretty important to you and I. There'll be, of all those millions of nerve cells in your brain firing all the time. Have you heard of them?
Yes.
You have, excellent. So neurotransmitters are really, really important chemicals. There's all sorts of different types of chemicals. You can go away and look at them, but some that you might recognize would be something like acetylcholine, dopamine. So these are all things, again, that stage 1 will go much more into depth about how they're working and what they're doing. Right, for now, you don't need to know that depth. You just need to know that there are chemicals that we call neurotransmitters. And these neurotransmitters can be excitatory. So if we have a neurotransmitter here, once a signal, an action potential comes along, sends a signal here, and basically what it will do is cause the neurotransmitter to be released into this cleft. And in here, you can see we have something called a receptor. So a neurotransmitter is a chemical, and a receptor is specific for one type of chemical. It will only respond to that one particular type of chemical that it's meant to respond to. So if the wrong neurotransmitter comes along, it will do nothing to this receptor. Now, if the right one gets released in here and binds to the receptor, it will pass the signal on to the next cell. And that signal can be excitement. So it could be that it needs to fire another action potential. So sending the signal on, firing another action potential. There can be cases where actually you want to inhibit another signal happening. Like, for example, in the huge networks in our brain, you might want to stop another signal from going somewhere. So sometimes these are inhibitory as well. And that would cause that hyperpolarization, that extra dip on the curve that we talked about. And some of them are both, just to confuse matters. But mainly the ones we talk about are excitatory. So really all you would need to know at this stage is that our synapses, they use neurotransmitters. Those are chemicals that bind to specific receptors and pass on the message. And that's how all that interconnection will be happening within our brain. Okay, so here we go. We've got an example of that here that looks a bit more specific, because the other one was quite a basic picture. So it involves several steps. So here we go. We've got that neuron, that presynaptic neuron, and it will be storing all the neurotransmitter in these little vesicles. So basically little balls of neurotransmitter all stored together. The electrical signal comes along, causes that to be released, and that will then bind to these specific receptors that can only recognize that chemical, and that will give you a response. We do then not want that to sit around. If we had neurotransmitter released and it just sat there all the time and didn't ever go away, then we'd be back in that same problem, that our nervous system would be continuously activated. And we don't want our nervous system continually activated. That's no good to you and I either. Sometimes it already feels like you have brain overload, let alone having everything constantly firing. So what needs to happen is within your nervous system, there needs to be a way to get rid of this neurotransmitter, so that when the next signal comes, it can be released again, but so that it's not constantly there. So there's a few ways that can happen. Basically, it can get taken back up and stored again. So in some systems, you can get some of it taken back up and it gets stored again, ready again. But we do have these other cells. These are like the support system for your nervous system. They're like the cheerleaders of your nervous system. So there are actually many more of these glial cells. And again, at stage 1, we're going to cover all of the types of these. And they are there to support your nervous system, to help it. They protect it. They have immune function, all sorts of things. But one of the other things they can do is help to break down neurotransmitter and get rid of it, so it's not stuck there all the time. So yeah, hopefully that will be so hopefully that will be a way of our body controlling this. Sorry, an email managed to pop up even though I've turned that on, so apologies for that. It confused me for a second. Okay, so the reason I wanted you to know about neurotransmitters and chemical transmission is I wanted to talk to you about some of the more exciting stuff in this. So my question was, or is, do you know of any drugs that you think might act on your nervous system, that might be acting on this, that might be causing problem with neurotransmitters? Can you think of any type of drug that might do that?
Maybe heroin and those type of drugs.
Yeah, absolutely. Well done. And as simple as nicotine, so smoking, and alcohol, caffeine, all of these things are actually acting there. So here we go. I've got quite a few here. Sorry, there's lots of words on this slide, but I think these are quite exciting and interesting for us to learn about. Not only because, one, we need to have quite a good public health education about this, but also as scientists, some of these things we can take advantage of and start to understand and use as drug mechanisms as well. And some of these are relevant, for example, in Parkinson's disease therapies, and that kind of thing. So I have got the name of some neurotransmitters in here. Again, don't worry about them too much. I've put them there because it would have looked really confusing without, but you don't need to worry about what the specific names are too much. It's just to explain to you what they're doing, really. So we've got cocaine. So I suppose you were thinking along the lines of heroin, that kind of thing. What that does, it stops the uptake of a neurotransmitter called dopamine. And dopamine is excitatory, so it can't be taken up. So like I said to you, it gets stuck in that synaptic cleft, and it's basically making that synapse constantly be on and activated. And that's why you get that wide-eyed look of somebody who's taken cocaine, because their nervous system's literally switched on by that, and that's why they get that sensation of a rush. And of course, over time, that becomes addictive because the numbers of receptors in the synapse starts to, your body knows this is wrong. Well, we shouldn't be switched on like this all the time, so I'm going to try and adapt to stop this from happening. And it starts to change some of the numbers of receptors to stop it happening. So then you need more and more cocaine to make that excitatory rush happen, and this is how addiction can happen. Similar with amphetamines, that's another type of illegal drug that might be taken. So a similar thing, that you have neurotransmitters released, so you're constantly on. But people forget, even smoking, of course, we know smoking is addictive. We know smoking is not good for you in many, many ways. And actually, that's mimicking the shape of a neurotransmitter called acetylcholine. And so it binds to the receptors for that in the brain, and then is mimicking that. And again, it has similar ways of becoming so addictive. This one's really interesting, Cura, because it has been used in medical terms as well, so in anesthesia type terms, but it can block acetylcholine receptors, so they can't be activated, and it causes a paralysis. So again, it's blocking the receptors, this one, instead of stopping it being reuptaking or stimulating or mimicking it, it blocks. So they're all acting a bit differently. They all do slightly different things there, but in the end, they're all causing the nervous system to be on or off when it shouldn't be. Similar for nerve gases. And this is the one drug of choice that I have, caffeine. I can't live without my coffee in the morning. Probably means that I am addicted to it. In fact, I gave it up for Lent once and got terrible headaches. So it causes release of calcium ions within the cells and makes action potentials be fired more easily. So because they're fired more easily, it's having that effect of waking you up that caffeine has. And alcohol, in fact, is doing the opposite, and it inhibits release of neurotransmitters. So there's fewer things activated. So yeah, all these things, not great for you. And part of the reason, I mean, alcohol, you only have to look at the hangovers people would have to know that's not great for you. But as well as all the other effects they're having, then they are having these effects directly in the brain. Okay, that's me blasting you with some biology that is hopefully some foundation to help you next term. And then I thought perhaps I can tell you a bit about my research, because that's what you're coming to do, I guess, is to learn more science. And so I won't tell you too much about it, but I'll just tell you a little bit more about my cold shock research and the work that I've done. This is with Professor Mark Smales as well. I've worked with him for a long time, so you'll see quite a lot of our stuff is joint. So what are your thoughts about what's persuaded you to take a biomed degree?
So the main reason was like I liked chemistry and I also liked biology. So I thought that a degree in biomedical science would like cover both those areas. So
Yeah.
that was one of the driving forces. And I also thought, like, I've read a few articles about research in biomedical science, and I found those interesting. So, and it's also a good way to help people behind the scenes. So that was more of the reasons.
Yeah, fantastic. And that's my reasons, really. I knew that I didn't want to be a medical doctor because I'm not that great with gore and blood and things like that. But I knew that I wanted to do something that would help people. And so that's why I went initially into biochemistry, and then I went and did pharmacology. Actually, that was quite gory. I had to get bits of heart tissue from patients who were undergoing surgery, and then I would test drugs on that once it was removed from their body in organ baths. And then I thought, okay, I want to do something that will really make a difference therapeutically. And this group, Mark's group at Kent, they were working on making antibodies cheaper. So certainly here in the UK, but I guess we've been highlighted, haven't we, by COVID, what healthcare inequalities there are in the world, and they're massive, and it depends on what each country can afford. And if you're making monoclonal antibodies, they cost around £3,000 to £5,000 a course often. So many countries can't afford to provide those for their populations. Many people wouldn't have insurance that would pay for it. And even here in the UK, where we have the NHS, some people would get access, some wouldn't. So they were working on that and were quite interested in making those antibodies. And I was quite interested in how we could do that and make it cheaper, because I'm quite interested in healthcare inequality. And that led, while we were doing that, one of the ways you can make more of these antibodies in cells is to make the cells cold and cold shock them. And meanwhile, another group worked with the professor I was working with, and they came along and said they were quite interested in this for dementia. And so they're quite interested in this from looking at mice who have models of dementia, so prion disease, so it's causing them to have dementia-like symptoms. And one thing that we'd all noticed is that this particular protein, when you cold shock the cells, it goes up, but it also might be protective for these animals. So like I said at the beginning, we kind of thought, we know that the cold can protect people's brains, but why? We can't go and subject elderly people to hypothermia. It would be really unethical. But if we can find the pathways and work out the science of why does the cold protect your brain, then you might be able to induce that pathway chemically without ever having to make people cold. And that's kind of what the group's been working on, and what I've been working on in my last sort of research projects would be to look at how, when we call things, this changes the proteins, and could we manipulate these proteins and basically use them as drug targets. And we've been working with a group, well, we've been working with a couple of groups in Leicester and in Cambridge on this. And really cool, the people in Cambridge actually have a group of cold water swimmers. So these are people who like to go to the Lido in London, and no matter what the weather, they like to jump in the cold water and swim. It sounds like my idea of torture. I can't think of anything worse than jumping in freezing cold water. And of course, you have to be really careful. You can't just go jump in freezing cold water if this isn't something you're used to doing, because it can cause quite dangerous responses. But these are people that do this all the time anyway. And they had a look, and these people actually do have those proteins upregulated. And we also know that there are other groups of people who do this cold water swimming, who've been a bit protected from dementia. So now that we know that this is a real thing, that in real humans, not just in cells, these proteins get upregulated, then now we're trying to figure out a way to target this with drugs, and we're working with those groups to do it. So it's really, really like cutting edge science that's making BBC News. So really exciting science. And hopefully sorry, carry on. Yeah.
It sounds really exciting.
Yeah, honestly, some fantastic things going on here. We have another lecturer who's just been looking at mesh code theory and how this relates to how memories and information are stored in your brain. Absolutely fascinating research on the cutting edge of the nervous system. So some really great things that are sort of going on in this area.